Um, okay, so Eurofins, as you may or may not know, is a large multinational company with over 60,000 employees working in over 900 laboratories in 61 countries. And this year marks the 35th anniversary of Eurofins. And it all actually started with a single wine testing assay back in 1988. And back then there was only 3 employees, but guess what? The 3 employees got lucky. It turned out that the single NMR-based wine testing assay turned out to be very useful. And it is still the gold standard for testing sugar additives in drinks today. So along the way, Eurofins expanded very rapidly into various testing business, including food testing, environmental testing, consumer product testing, pretty much anything you can think of. It wasn't until the year 2011 that Eurofins emerged as a leader in biopharma services after the acquisition of several key companies. So Eurofins Discovery is a small part of the entire group, obviously. It's a network of 11 laboratories in 6 different countries, and each site has its own field of expertise. And together, these laboratories, they complement one another to cover the entire scope of drug discovery. Whenever a project requires integration of multiple specialties, there will be a global project committee that works with the client to determine the best technologies or the most efficient path moving forward. So all in all, you can view Eurofins as a network of resources that will provide our clients with comprehensive end-to-end solutions, unique platforms, as well as fully customizable services. Okay, so the Biotherapeutics division is a relatively new chapter for Eurofins Discovery. It was actually first initiated in the year 2020. And the team is led by Dr. Fran Niethling. She spent almost a decade at Boehringer Ingelheim before she joined Eurofins. And she's accumulated over 20 years of experiences in drug development. And she's very resourceful and knowledgeable. And then some other key members of the APAC team include Elsa, who is the guru of all cell-based assays, and Kai Lin, and this is me. Kai Lin and I, we have similar backgrounds in antibody drug development, so we oversee various characterizations. And I myself have worked on non-clinical development of novel protein drugs and several biosimilars in the past, and I was involved in IND filing and some discussions with the FDA or EMA. And we also have Dr. Judy Fu, who specializes in structural biology. Okay, so this is kind of a busy slide. What it's meant to show that is, as a global team, Eurofins can cover almost the entire drug development process as a one-stop service provider. As you can see, we'll provide many catalog as well as custom assays during each phase of the drug development process. We also work with carefully selected strategic partners on certain platforms. And so today I'd like to walk you through some of our capabilities that are most pertinent to building a strong IND package. So first, for target identification, we offer proteomic analysis. As you know, proteomic analysis has wide applications during different phases of the drug development process. It can be used to verify a drug's MOA. It can also be used for identification of biomarkers that are associated with certain indications. But most importantly, it serves as an unbiased way to identify potential new targets for or new drug targets for therapeutic purposes. So the general workflow for proteomics goes from sample processing to protein digestion, and then data collection and analysis. So you can see in this example below where cell extracts from treated groups were collected for analysis, but we can actually process animal tissues as well. So some clients have asked us to help validate their animal models this way. So after the samples in this case study were processed, you can see in this volcano plot that the levels of some of the proteins highlighted in the red box were increased, but more clearly is you see decreased levels of many more proteins, suggesting that the drug treatment resulted in a decrease of these proteins. So after we collected the data, we will also analyze them and annotate these proteins and kind of categorize them in different areas by function in these graphs here. Okay, so once a target has been identified and validated, maybe the next step would be to generate therapeutic antibodies against the target. Towards this end, Eurofins offers access to this state-of-the-art phage display library for generation of human antibodies. So this is a SCFV synthetic library of the size 10 to the 11th. So in this library, the SCFV sequences, they were designed to capture the entire human immune repertoire, like shown here. And these SCFV sequences, they were then fused to the viral code protein so that when the viral particle is expressed, in theory, there should only be one SCFV molecule associated with that viral particle. And whenever this SCFV has binding interaction with the target protein, then the genetic information can be retrieved by sequencing the phage mate here. And this process is usually repeated 3 to 4 times to enrich for binders. So this is the overall workflow for phage display panning. What's special about our library is that there is no milestone or royalty payments associated with using the library. It's operated solely on a fee-for-service basis. And also, our clients would have full intellectual claim over whichever sequences they discover during their process. And as you'll find out soon enough in the later slides, that we have a wide range of protein production and characterization options available to better support your research program. Okay, so the library of the phage display library is usually assessed by performing panning with the library. In this table here, it shows data from panning against 6 different proteins, and an average hit rate of 60% was obtained across all 6 different proteins. But more importantly, a significant number of unique clones were obtained for each of these target proteins. This is important because obviously we don't want the positive binders to be all the same. We want them to be as different as possible, so then we have more options to choose from. So when these antibodies, they were converted for full-length recombinant expression, and they were characterized for a binding affinity by SPR, their KDs were found to be in the range of sub-nanomolar to nanomolar concentration range. And this is a good range for most of the applications and it's also in line with some of the best or the well-known phage display libraries in the world. And to the right, you'll see an example of one of the anti-TIM3 antibody that was discovered in this library. So it was tested in this reporter cell line assay, where blockade of TIM3 signaling result in decreased signal output in this assay. And to the right, you'll see that the antibody generated from this library has over 100-fold better potency than the commercial control antibody 2E2. So in addition to phage display library, we also offer a hybridoma platform for generation of reagent antibodies as well as anti-EDO type antibodies. What may differentiate our platform from others is that our facility is AAA LAC and OLOG certified, and all the procedures in hybridoma generation, including immunogen design, immunization, and screening design, they're all fully customizable. In addition to that, our pricing is competitive, and since all the procedures are done on site, excuse me, at our facility, there is no confidentiality risks associated with this process. And so in order to generate monoclonal antibodies, we usually start with immunogen design. For peptides and small molecules, they require conjugation to the carrier protein in the beginning to increase the immunogenicity. For other protein entities, we would make sure that the proteins are properly folded. And then following immunization and fusion, we can then conduct a series of ELISA screens, both positive selection and negative selection ELISA. And if it's an anti-EDO type antibody, we will also determine if the anti-drug antibody is blocking antibody or non-blocking antibody. So once we selected the hybridomas with the properties that are desirable, then we can sequence the hybridomas and convert them for full-length recombinant expression. And once again, all the procedures in this process can be tailored towards our clients' requirements. Okay, if one decides to generate therapeutic antibodies through this hybridoma technology, we can also offer antibody humanization service. Our team is highly experienced in the CDR grafting method to produce the humanized antibodies. So in this process, we first take the CDR sequences and we engraft them to the most homologous human framework. And then we design 5 back mutations on the heavy chain and on the light chain respectively. And together they create a total of 25 back mutation variants. And then we express those 25 variants, and we characterize the KD by SPR. So then the final humanized antibody is guaranteed to have comparable affinity to the original mirroring or the chimeric antibody. As you can see in this table below, where 10 of the antibodies humanized in the past all had comparable affinities towards the original chimeric antibody. This is a ratio comparing the KD of humanized antibody to the chimeric antibody, and they're all threefold. And that's why we consider it as comparable. It is guaranteed that we can humanize these mirroring antibodies. Okay, so next I'm going to talk very briefly about our protein production capabilities. We have 4 platforms for transient recombinant protein production. We have 2 mammalian cell lines, including XP293, which is a human cell line, and XPCHO, which is a hamster cell line, as well as the insect cell line, XPSF9, and E. coli recombinant production platform. And all 3 of the XP cell lines are well known in the industry to be very robust and high yielding. As for timelines, in general, it takes as little as 4 weeks to go from gene synthesis to purified proteins. We have a number of these expression constructs ready, so your gene of interest can be expressed under different conditions simultaneously in order to save time. And then the quality of these proteins, they're usually assayed first by standard QC procedures such as the capillary electrophoresis. This is the chip-based CESDS, as well as SEC-HPLC for purity. And then there is a range of additional characterizations that are available to verify the protein quality, such as the intact mass, glycan profiling by HPLC, ELISA binding, of course, and as well as binding characterization by SPR. Okay, so as the antibody therapeutics keep evolving in this field, more and more of these unconventional formats, such as the multi-specifics or the antibody fragments, have entered the pipelines or even clinical space. We at Eurofins have also accumulated some useful tools or purification schemes that will help increase the overall yield of these more challenging antibody formats. And when the yield is not optimal, we don't just stop there and leave that to our clients. We will actually go back and work with our clients to see if we can increase the expression or try to adjust the purification scheme to try to increase the overall yield. And that's when some of our expression constructs come in handy, because we can convert between these formats very rapidly in order to get sufficient quantity of some of these more challenging formats. Okay, so now I'm going to switch gears and talk a little bit about various aspects of protein characterization. So when we're working with a new drug molecule, it's important to conduct a very thorough characterization as early as possible in order to determine the critical quality attributes, which are properties of the protein that would likely have an impact on safety or efficacy of the drug candidate. So towards this end, excuse me, we have a series of catalog assays that are based on LCMS or UHPLC to characterize the antibodies. So we know that post-translational modifications is very important for proteins, and it's one of the most defining property of a biosimilar molecule. It will decide whether the biosimilar molecule will be successful or not. I actually came across this biosimilar molecule with a unsimilar glycan profile that ended up being pulled out of the program after phase 1 clinical trial due to a unsimilar PK profile. So apparently the unsimilar PK was due to the unsimilar glycan on the protein. So the story tells us that it would be best to characterize the critical quality attributes as early as possible in order to make your research program more successful. So understanding the binding properties of a drug candidate is also one of the most fundamental characterizations that need to be carried out for a drug candidate. And at Eurofins, we have a dedicated SPR team that would characterize both large and small molecules binding. We have many catalog assays available, already established for antibodies, but we can always do method development for any pair of molecules of interest to our clients. More importantly, the BioCore instrument is AK+, is BioCore AK+, which is a high throughput model. It's very suitable to run a large number of samples. And in the case for biosimilar studies, it works really well because in this case, you would have to compare multiple lots of samples in parallel. So shown to the right is an example of that. You'll see different batches of the originator Opdivo compared with different lots of the Opdivo biosimilar. And you can even demonstrate the biosimilarity by doing this parallel line analysis, PLA. And we can also carry out non-GLP qualification for lot release purposes if necessary. Okay, so next, I'm going to talk a little bit about in vitro functional assays. We all know that the in vitro functional assay required for each drug candidate would be dependent on its MOA. And that is why we have a large number of assays already established for our clients to choose from. In fact, we have over 220 of these cell-based assays already validated. And many of these assays are aimed to measure how the drugs affect certain aspects of the cell physiology. There are also assays that focus on particular molecules like GPCR, NHR, or they can focus on a particular signaling pathway like the calcium influx. But regardless of the nature of these cell-based assays, the output are usually measured by either the plate readers or the high content confocal imager, flow cytometer, or Luminex. So our latest tumor profiling panel would be a good example of the type of cell-based assays that we run routinely. In this panel, we selected 36 tumor cell lines that cover 13 different cancer types. So a quick run of your drug candidate in this panel would tell you a little bit about its efficacy towards different indications, perhaps. So once you have these data from the in vitro screen, our clients can transition very smoothly into the in vivo animal models because all 36 corresponding xenograft or syngenic models have already been established at Eurofins. So then our clients can select the most favoring animal model easily based on the in vitro screening data. Okay, so on this page is a list of the 36 cell lines for in vitro functional assays, as well as in vivo animal models. And we also have additional cell lines that have been validated in vitro, but don't have the in vivo animal models established yet. But we can certainly customize and run these method developments if necessary. So I just have a couple more examples more relevant to oncology indications to share with you. So first, this is a 3D cell model for angiogenesis. We all know that HUVEC cells are notoriously difficult to work with. In this example, we culture the HUVEC cells with the 3D matrix, and then we quantify tube formation. It's the fluorescence you can see in this panel here using confocal or high content confocal Z stacking. And you can see that the tube-like structure start to disappear as we increase the concentration of the positive control drug sumarin. So this will serve as a robust assay for screening anti-angiogenic compounds. Another example would be the examination of Fc effector functions for antibodies. So depending on the isotype and the possible mutations in the Fc regions of the antibodies, there may or may not be ADCC, CDC, or ADCP effects. So in this example here, we demonstrated the ADCC effect of a Herceptin biosimilar. In this experiment, the PBMC was incubated with fluorescently labeled cancer cells. And when you add the antibody, you see formation of these immunocomplexes and disappearance of the cancer cells because they're dead. And we have actually a couple other alternative ways to do this in order to have lower background, even lower background for these target cells. We can use luminescence instead of fluorescence. We can also use our proprietary killer cell lines by DiscoverX as the target cells in these assays. And very similar setup, we can incubate human sera as a source of complement with these target cells I just mentioned to measure CDC effects. As for ADCP, we currently use a engineered reporter cell line. Okay, so next I'm gonna switch gears and talk a little bit about structural determination. So many people may have the impression that only academic groups are interested in solving structures, or that structures are not very useful in the industry because it's not required by the FDA. Well, it is true that structure is not required by the regulatory agencies, but at the same time, ever since cryo-EM emerged as an important technique for solving structures, actually more and more companies are now interested in having structural determination as one of their standard characterizations for new drug development. So for a new drug, getting the structure would help structure-based drug design. As shown on the right here in this diagram, once you have a hit, you'll be able to perhaps solve the structure between the drug in complex with its target. And then you can in turn use that information to come back and redesign or to do modifications on your drug to enhance for affinity or better selectivity and so on. So you'll be able to perform structure-based drug design. And also, structural information would be key to add value to your projects, especially during fundraising, and also interactions between the drug and its target would help secure patents. And finally, statistics show that drug development programs with structures solved are more likely to get approved. Okay, so Eurofins offer both cryo-EM-based and X-ray crystallography-based structural determination. Cryo-EM is a method that's more suitable for large protein complexes or membrane proteins. And the quantity of protein that's required for this technique is very little compared to X-ray crystallography. As low as 0.2 milligrams of protein is enough for cryo-EM. And the purity is usually greater than 90%. And the reasonable resolution that you can obtain with cryo-EM nowadays is about 2 to 5 angstroms. And once the protein is ready, it takes about 6 weeks to go from the protein to structure. Excuse me. So for X-ray crystallography, it's more suitable for smaller proteins. And in contrast to cryo-EM, it requires a relatively large amount of protein, 5 to 20 milligrams. And the protein has to be stable enough at high concentrations so it doesn't precipitate before it forms crystals. And the purity is usually similar to what would be required for cryo-EM. And the resolution you would obtain with X-ray crystallography is probably slightly better than cryo-EM. And since there's no way to predict whether crystals will form, it makes it very difficult to predict how much time it actually takes to solve structures by this method. However, if they're homologous proteins that have their structure solved by either of these methods, then chances are similar procedures will likely work for your protein. Okay, so this is the last section of the talk. I'm going to talk a little bit about our in vivo part. So once the drug candidate has been tested thoroughly in vitro, the next step may be to test it in animal models. And our in vivo pharmacology team has tremendous experience in over 350 efficacy models in 6 different therapeutic areas shown here, plus PK/PD and non-GLP safety. Okay, so just a list of our oncology animal models we currently have established. There's a total of 46 xenograft models, 10 syngenic models, 9 orthotopic models, and 2 PDX models. For anything that our clients would like to establish but are not shown on here, it would be possible to do customized animal model establishment. Okay, so I just want to share a couple examples of our oncology models here. To the left is a human xenograft model, colon cell line SW480, and then to the right is a murine colon syngenic model. I'd like to show you that both of these models, as well as most of the other models I showed on the previous page, have been extensively validated with known large and small molecule drugs. So this kind of data would usually give our clients a sense of how much to dose and what kind of outcomes to expect. And then our latest addition to the oncology animal model capability would be live cell imaging with IVIS spectrum. In this case, obviously, it wasn't live imaging because the animal model was dissected to look at metastasis of these cancer cells. when combined with the orthotopic mode of injection, it can be a really powerful tool to look at metastases of cancer cells that are being labeled with luciferase expression. And some of the data actually correlate pretty well with actual clinical data. Okay, so once the primary efficacy study is completed, some of our clients may be interested in the ex vivo analysis. For example, measurement of the level of cytokines by Luminex or tumor infiltrated lymphocyte analysis by flow cytometry. There's a whole range of other ex vivo analysis that we can perform on site. Okay, finally, our animal team also has some experience with RNA drugs. And in such case, we can also help with the mRNA production, starting from the in vitro transcription. By our standard methods, our client only needs to provide a sequence and will be able to produce the mRNA with the same standard as the vaccine, the current vaccines. And then we can also do encapsulation of the mRNA molecules using some of the more commonly used Moderna formulation or the BNT formulation or any combination that our client specifies. And a 95% encapsulation rate is guaranteed. And once these is all done locally, we can guarantee integrity of the LNP formulated RNA molecules, and we can inject into our animal models for efficacy or more validations. Okay, so that's the end of our presentation, and I'll be happy to take any questions.